Hello everyone. Today we're going to be talking about cytokines and chemokines. These are things that you will have heard mentioned in many of your previous lectures, but today we're going to go into them in a little bit more detail. Before I begin, this is just a reminder that all of the figures used in this presentation can be found in your textbook. There's going to be a lot of jumping around in this particular lecture, so I would encourage you to pay attention to the bottom of the screen, either the left or the right, depending on the slide. That'll tell you which figure we're on and where you can find it. The first number would be the chapter, the second is the figure number. Today we will have 4 objectives that we'll go through together. The first of which will define cytokine and learn about the different families and receptors, including the interferon family, which you may have already heard a little bit about. Second, we'll define chemokine and distinguish between the 2 types of chemokines, Cc Chemokines and Cxc Chemokines. For our third objective, we'll learn to understand the importance of cytokines and chemokines in mounting an inflammatory response. And finally, we'll briefly talk about the 3 different types of interferons. So let's begin with defining cytokine and talking about the different families of receptors of cytokines and cytokine receptors. So what is a cytokine? Well, a cytokine is a small protein that weighs about 25 kilodaltons. If you break down the word, cyto means cell and kine means movement. This suggests that the main role of a cytokine is to cause movement and change within cells, in our case, specifically lymphocytes and other immune cells. Cytokines can act in 3 different ways. They can act in an autocrine manner, meaning the cell that produces a cytokine will also receive the function of the cytokine. It can act in a paracrine manner, which means that once a cytokine is produced, it will act on neighboring cells or adjacent cells near the producer. And finally, it may act in an endocrine manner, or on cells that are very distant from the producer or systemic in the organism. It's important to note that cytokines are grouped into families. This is based not only on their structure, but also on the receptors they use to signal. So there are 4 main types of cytokine families, the first of which is the IL-1 family. You'll probably have heard a lot about this when talking about the innate immune response. These include IL-1 alpha, beta, and IL-18. These are all products of the inflammasome, something you might remember from the innate immune system lectures. The second family is the Hematopoietin family of cytokines. This includes IL-6, GM-CSF, IL-2, and IL-4. We'll talk about a few of these in more detail today. The third family is the TNF family, which consists of TNF-alpha and others. And the final family is the Interferon family, which notably consists of interferon alpha, beta, and gamma, but there are other interferons as well that we just won't talk about today. Each of these cytokine families signals through a certain type of receptor, which we'll go through now. The first type of receptor that cytokines may signal through is through a homodymeric receptor. It's important to note before I continue that all of these receptors are dimeric and have 2 components. However, in this specific case, the 2 dimers are exactly the same. These type of receptors are typically used for erythropoietin and growth hormone, 2 endocrine cytokines. Another type of receptor that you may have is a heterodymic receptor that shares a common chain. This common chain means that many different types of receptors may contain one component of the overall receptor. IL-3, IL-5, and GM-CSF share a common chain, CD131, while the second component of the chain is different between the 3 cytokines. Receptors for IL-2, IL-4, 7, 9, and 15 also share a common chain, CD132, while the second and sometimes third chain differs between the group of them. Another possibility for receptor is the heterodymic receptor that has no common chain. This means that the 2 dimers or components of the receptor are different, but neither of them are shared between different families or different types of cytokines. This includes the IL-1 family of receptors and receptors for IL-13, interferon alpha, beta, gamma, and IL-10. You also had the TNF family receptor, which recognizes TNF, of course, and also interacts with CD40, FAS, CD30, CD27, and nerve growth factor. Finally, you had the chemokine receptor family. This one is a little bit different than all these other ones I've previously discussed. All of these upwards are what we call receptor tyrosine kinases, which means that they rely on phosphorylation of intracellular components of the receptors. However, the chemokine receptor is a little bit different. This is called a GPCR, and we'll discuss it in a further and later objective. As I mentioned before, many of the cytokines signal through shared receptor components. A good example of this is IL-12 and IL-23. Both of these signal through one component of the receptor, IL-12 receptor beta 1. However, the second part of the receptor that makes up the dimer is different between the two cytokines. IL-12 uses the IL-12 receptor beta 2 component, whereas IL-23 uses the IL-23 receptor component. This allows for downstream functions to be different between the two cytokines. Although the receptor IL-12 receptor beta 1 is shared, the addition of the second different receptor allows for IL-12 to induce STAT4 transcriptional activity and IL-23 to induce STAT3 transcriptional activity, and thus result in differential functional effects. In the case of IL-12, you get the differentiation of Th1 cells, and in the case of IL-23, you get the differentiation of Th17 cells. So clearly, the receptor through which the cytokine signals is very important for determining the downstream functions. Once you are actually inside the cell, many of the cytokine receptors signal in a very similar way. We call this signaling pathway the JAK-STAT signaling pathway. This is usually mediated by a kinase, JAK or Janus-associated kinase, or STAT, a transcription factor that can go in, bind DNA, and cause the expression of different types of genes that are specific for the response you're looking for. So what happens first? Well, the dimers of your receptor are usually not associated on the surface of the recipient cell. They're separate, they're floating, and they can't recognize each other until they've been bound by a ligand, or their cytokine. Once the cytokine binds the receptors are then able to dimerize and allow their JAK regions, or their intracellular components, to come together. Once the JAKs are in close proximity, they can activate each other and phosphorylate the ends of the receptor. This phosphorylation occurs on a tyrosine residue and will allow for an SH2 domain that's part of the STAT transcription factor to recognize the phosphorylated receptor. Binding of the STAT transcription factor to this phosphorylated site on the receptor will change the conformation of the STAT transcription factor and allow it to dimerize with another STAT transcription factor. This STAT dimer will then translocate into the nucleus and cause the expression of different types of genes specific for the necessary response. This is everything I've just said, listed in a little bit more organized manner. We'll go through it one more time just to belabor the point, and then we'll move on. In the case of the Hematopoietin family of cytokine receptors, you use the kinases JAK1, JAK2, JAK3, and type 2. These come in different combinations depending on the type of receptor that you have in that context. When the JAKs are brought close together by the dimerizing cytokine receptors, they'll phosphorylate the opposite receptor on tyrosine residues on the receptor itself. A phosphorylated tyrosine, as you may remember from earlier lectures, can be recognized by a conserved SH2 domain, which is located on many different types of signaling proteins. In this case, the SH2 domain that recognizes the phosphorylated tyrosine on the receptor belongs to the STAT family of transcription factors, or signal transducers and activators of transcription. STATs that associate with that SH2 region or that phosphorylated tyrosine region on the receptor then also become phosphorylated. The phosphorylation of the different stats allows them to dimerize, allowing them to form stable structures that can then translate into the nucleus and initiate transcription. This signaling pathway is incredibly important for organizing all of the different inflammatory responses that are necessary when our organism or the body faces an outside threat. Many of these you'll have heard before, and some of them we'll talk about again today. IL-1 beta, TNF-alpha, and IL-6 are immediate responses to infection and are important in mounting an inflammatory response at the beginning of the infection. We'll talk about this in much more detail in one of our later objectives. Some cytokines we'll spend less time on today, but are still important to know, include CXCL8. This is actually a chemokine, which means that it allows for the migration of cells towards the site of infection, and is important for recruiting neutrophils, or the first responders, when your body sees tissue damage or an invading pathogen. Another one is IL-12. You'll have heard a lot about IL-12, especially in the context of T cells. It's responsible for skewing the T cell differentiation into Th1 and is produced as an effector cytokine by Th1s as well. This brings us to our second objective for today, which is to define chemokine and distinguish between the two types of chemokines, Cc and Cxc. So what is a chemokine? A chemokine is technically a type of cytokine. It acts as a chemotaxin, and it causes cells to migrate towards a site of higher concentration. This means that as a chemokine is produced, the amount of chemokine at the site of production is much higher than further away. This allows migrating cells to get closer and closer to the site of production while following a line of increasing concentration of the chemokine. Chemokines have two main functions. The first of which is to cause a conformational change in integrins, allowing leukocytes migrating towards the lymph node or site of infection to stop rolling across the endothelium and instead perform diapedesis and migrate through the epithelium into the site of infection. The second function of chemokines is to direct the path of migration with increasing concentration gradients, something we just talked about at the beginning of this slide. It's important to know that chemokines are ubiquitously produced by both immune and non-immune cells. So how does a chemokine signal? As I mentioned earlier, the chemokines actually signal through a different type of receptor from other cytokines. This receptor is called a GPCR. Some examples of what it recognizes are some examples of the receptor itself are CCR1 through 10, CXCR1 through 5, XCR1, and CX3R1. Chemokine receptors, as I mentioned, are called GPCRs, or G-coupled protein receptors, meaning that they use specialized G proteins in order to transduce their signal. We'll go through that now. So what happens when a GPCR sees its signal? Well, before the ligand comes along, or the chemokine comes along, the GPCR sits unassociated with the G proteins that will allow it to signal later on. At this time, the G proteins are also bound to GDP and are in an inactive state. When the GPCR binds its ligand, or its chemokine, it then is able to associate with the nearby G protein. Once this occurs, the G protein can release GDP and instead bind GTP. This essentially activates the G protein to signal. Once the G protein is bound to GTP, it will then dissociate into two components, the alpha component and the beta-gamma component. These two components will signal through molecules like RAC and Rho, CDC42, to cause the chemotactic motion of the cell or the migration of the cell towards an increasing signal concentration. Once this occurs, the GTP on the alpha subunit is hydrolyzed to GDP. This inactivates the signaling of the G proteins and allows them to reassociate and dissociate from the GPCR until it sees another ligand or its chemokine again. There are two distinct types of chemokines or cytokines, the Cc Chemokine and the Cxc Chemokine. These differ not only in their structure, but also their general function. Cc Chemokines have two adjacent cysteines near their N-terminus. Recall that cysteine is an amino acid that makes up the component or the protein structure of the chemokine. Cc Chemokines typically promote the migration of monocytes, lymphocytes, and other cell types. A good example of this would be CCL2. CCL2 acts on monocytes through the CCR2b receptor on the surface of the monocytes and induces them to migrate towards the tissue and become macrophages when there's an infection. Cxc Chemokines differ only a little at their N-terminus, and that the two cysteines are separated by one additional amino acid. The X means that it can be any amino acid, and it's usually not a specific one. Cxc Chemokines, in contrast to Cc Chemokines, typically promote the migration of neutrophils to the site of infection. An example, as we mentioned earlier, is CXCL8. This will be the first chemokine the neutrophil sees upon infection and will allow the neutrophils to accumulate at the site of tissue damage or pathogen invasion. So let's go through a couple examples of chemokines, and then we'll talk about some important ones a little bit later. One of them we've already mentioned. This is the CXCL8 that causes the migration of neutrophils to the site of infection. Another one is CXCL7. CXCL7 is produced by platelets, which means that the resulting function of CXCL7 signaling will ultimately benefit something that the platelets have sent specifically. This activates neutrophils, causes clot resorption, and angiogenesis. These are all important for maintaining our blood vessels and the integrity of our circulatory system. Some other examples are CXCL1, 2, and 3. And while they do signal to other immune cells, you can see that, like the other Cxc chemokines, they also are responsible for recruiting and activating neutrophils once they've reached the site of tissue damage. Some examples of Cc cytokines include CCL3 or MIP1 alpha, which is responsible for recruiting monocytes, NK and T cells, basophils, and dendritic cells. This is important in antiviral defense as well as Th1 immunity. Another example is a closely related member, CCL4 or MIP1 beta. It recruits many of the same cell types, but was evolved very specifically for helping to compete with HIV-1 infection. And finally, you also have examples of CCL2 and CCL5, which are responsible for recruiting a whole skew of different subtypes of immune cells and are really important in Th2 immunity and recruitment of basophils. That brings us to our third objective for today, which is to understand the importance of cytokines and chemokines in mounting an inflammatory response. This is a very big and busy graph right here. We'll go through some of them together. Many of them you've actually already heard of, but this is just a good way of summarizing the different functions of some of these really big cytokines that you'll hear a lot about if you pursue a career in immunology. Some of these we'll talk about in the future, like IL-2 and interferon gamma. Some of these you've already heard about, including IL-4, 5, and 13. You may recall that those three cytokines are important for mounting a Th2 response or a type 2 response, which is important in responding to parasites and mediating the allergic response. TNF, or lymphotoxin alpha, is also important for mounting that initial inflammatory response, and that's something that we're going to talk a little bit about now. So three important innate cytokines are cytokines that are involved almost exclusively in the activation of an innate immune response are IL-1, 6, and TNF-alpha. They're critical to mounting the response to infection, and without them, the organism fares very poorly when it's infected with an invading pathogen. This is because it was unable to make the physiological changes it needed to make in order to accommodate an immune response and activate the full breadth of inflammation. IL-1 beta, IL-6, and TNF-alpha have many different effects, some of which we'll talk about. They act on the liver, on the bone marrow endothelium, on the hypothalamus up in your brain, on fat and muscle, and also on dendritic cells to help promote the adaptive immune response later on. These are good examples of endocrine cytokines, or cytokines that will signal systemically and far away from where they're being produced in order to make key physiological changes in the organism when it's experiencing invasion by a foreign pathogen.